Hello, everyone. Welcome to my video tutorial for spatial data analysis. Today, I'm going to show you how to analyze CODEX data in Seurat. So, Akoya Bioscience is another company that has image-based spatial technology. So it produces multiplexed spatially resolved protein profile. Actually now, they changed their brand CODEX into Akoya PhenoCycler. So now lots of software you can use to analyze the data generated by Akoya PhenoCycler. We can go to the Akoya Bioscience website and have a look at the data format and also the available software for the data analysis. So let's go to the Akoya Bioscience website. You can see here is the software and the data information. You can see here the CODEX is now PhenoCycler. They changed the brand name. And if you use PhenoCycler to produce spatial data, normally the data file will be a QPTF file. This is a compressed data file. And Akoya Bioscience has its own software to analyze the data, and also they have a list of software that you can use to analyze Akoya CODEX data. You can see here is the list. Because most software are commercially available, you need to pay to use the software. If you go down to the end, you can use the CPath open source software to analyze the data. I'm not going to show you CPath software today, but I have a plan to make video tutorials for the CPath software. Once you process your data using any available software, you normally get the information for the cell coordinators and also the expression matrix for different proteins in each cell. So we can export those data to do analysis in R. For example, today I'm going to show you how to analyze the data in Seurat. So you can see on the Seurat online tutorial, they have an online tutorial to analyze the CODEX dataset from the human lymph node. So you can click this link to download the data. We can click this link. You can see the first file, the CSV file, is the file that we need to download for today's analysis. If you click the download, you will download this dataset. I downloaded already. We can open this dataset and have a look at the data information. You can see here, this is the CSV file. You can see the first column is the cell ID, and we have the information for the cell position and also the tiles position. Then from column J is the expression matrix for different proteins. You can see the first one is the DAPI to recognize the cell nuclei. And if we go further, you can see this one is the CD8 protein. If we click another one, you can see this one is for the CD3 protein. We have 28 protein in total in this dataset. If we go to the end, you can see we also have the T3 information because the cells in this dataset were connected by other software. So I'm going to use this dataset for demonstration. But if you want to practice the method using other CODEX dataset, you can go to the Human Biomolecular Map website. If you use this link, you will see available dataset. I can show you how to find the a CODEX dataset. You can see on the left-hand side of the Data tab, if you view all, you can see there are 128 CODEX dataset available. If you tick on this box on your right-hand side, all the data type will be CODEX data. You can see there are data for the small intestine produced by Stanford University. If we click the groups, you can see the lymph node spleen data set produced by the University of Florida. So you can click the link to download the different data set for practice. So now we can go to R and perform the analysis for the lymph node dataset that we downloaded from the Human Biomolecular Map website. So for Akoya spatial data set analysis, Seurat has a function called node Akoya. You can use this function to load the Akoya data and create a Seurat object. So we can tell R where I save the data, then we can load the data. Let's run this code. So you can see we have a Seurat object. It contains the expression matrix data and also the cell coordinates information from the image. So we can use the row names to have a look at the 28 proteins that were analyzed in this dataset. We can run. You can see here are the names for 28 proteins that were studied in this dataset. So we created the Seurat object. Next, we can run pre-processing and unsupervised analysis to cluster the cells. Because this is protein data, so the recommended normalization method is centralized log ratio based method. So for the normalization method, we set it as CLR. Then we can normalize the data. So after the data normalization, we scale the data. So if you perform the single-cell RNA sequencing data analysis, the next step for the standard Seurat workflow is to find the variable features. Because we have lots of genes in the single-cell RNA sequencing data, we normally identify 2,000 genes as variable features. But here we only have 28 proteins. So instead of identify variable features, we can set all 28 proteins as variable features for the analysis. We know the row names are the names for 28 proteins. So let's set all the proteins name as variable features. We can set it. Then next we can run PCA. After PCA, we run UMap. After UMap, we find the cell labels. So the last step for the unsupervised analysis is to find the cell clusters. So now the data is ready to visualize the cell clusters. We can use the dimplot function to look at the cell clusters. Let's run. You can see with resolution at 0.4, based on the expression of 28 proteins, the cells were clustered into 13 cell clusters. Because we only have 28 proteins here, they are cell type specific markers. So today, I'm not going to show you how to annotate the cell clusters. It is very easy to identify the cell clusters based on the protein information. So next we can use the image plot function to look at the cell clusters on the image. So we can run the image plot. So you can see we created the image and show certain cell clusters on the image. Following the analysis, we can use the image feature plot to look at the individual protein expression on the image. For example, we can use the feature plot to look at the protein level 1 expression. It is a mark gene for lymphatic endothelial cells. We can run the feature plot. You just need to set the features as the protein name. We can run. You can see the red color shows high expression of LYMV1 on the image. Because we only have 28 proteins here, you actually can use the image feature plot function to look at the expression pattern for all the proteins. I list all the proteins down here. We can use CD31, CD8, CD20, Ki67 as example to look at the expression for 4 proteins at the same time. Let's run it again. You can see we generated the feature plot for 4 proteins. As I said, we only have 28 proteins here, and I listed all the proteins here. I'm not going to run all of them, but you can practice if you want to see the expression pattern for other proteins on the image. And also in the beginning, I showed you how to find the protein's name because they are the row names for the dataset. You can find all the proteins' name from the row names. So okay, that's for the demonstration. Actually, even we produce the image, this is not the real image that was taken by the microscope. So if you want to see the protein expression pattern on the real image, you can watch my future video tutorial for the Spatial software. I'm going to stop here for today's demonstration. I hope to see you in my future video tutorials.